Okay, so unfortunately, the camera stopped while I was in the middle of lecture, and so I'm just going to pick up the lecture here where it left off when I was giving it earlier in class. So that you guys have the same content for the exam on Wednesday. So the light-independent reactions, again, take all of the materials that were made in the light-dependent reactions, or the light reactions, so the plants can construct a sugar from it. And really what that looks like is this. I've given you this equation before. The equation for photosynthesis is carbon dioxide, and we're going to take 6 carbon dioxides. We're going to add to that 6 water molecules. That's going to give you, after there's some biological magic that happens, some sugar, which is C6H12O6 plus 6 oxygen molecules. Now really what happens is the light reactions, they take water and they oxidize it. So this is an oxidation into oxygen gas, which liberates a bunch of electrons. It's an oxidation reaction because an electron is being lost from water, and we get oxygen gas from that. So through that process, we're going to make some electron carriers, NADPH, and then also because we're breaking bonds, we're going to be able to generate some chemical energy that we make into the form of ATP. And then the other part of the reaction, this is the dark reaction, we take carbon dioxide and we're going to reduce, so this is a reduction reaction, we're going to reduce carbon dioxide into C6H12O6, or sugar. So this is an abbreviated version of the chemical reaction for photosynthesis that I would like you to know. A more expansive one is this, and you'll notice the difference is there are 12 water molecules here, and then they're in the reactants, and then there are 6 water molecules as products. This equation, I've just taken the products away from the original reactants. So let's talk a little bit about light. Light's an interesting phenomenon. It occurs as both a particle and a wave. And the way that we measure the strength of light, or a photon, the strength of a photon, is by its wavelength. And wavelength is defined as, so photons, when they leave the sun, they move in a wave pattern, like so. And we define photon strength by wavelength. And a wavelength is defined as the distance between 2 peaks of a wave. Now, that determines not just the strength of the photon or the amount of energy represented by that photon, but it also determines the color of the light. And we know that there are multiple colors, right? You can pass white light through a prism and it diffracts the light and you get a rainbow color produced. But really, the energy represented by photons extends far beyond that violet to red light that you and I can see. Photons occur in very, very short wavelengths, like the gamma rays. So gamma rays have a very, very short wavelength. And with photons, the shorter the wavelength, the stronger that, or the more energy that photon represents. So very short wavelengths, lots of energy. Very long wavelengths, much less energy. So gamma rays are very short, which is why when we expose our DNA to gamma radiation, it can disrupt our DNA molecule and cause mutations. We also have the ultraviolet spectrum that is a very short wavelength and a little too short for us to see. But that's the reason why when we go outside during the summertime, we get sunburned, because ultraviolet radiation is physically burning our skin. It's disrupting cell membranes, basically, in our epidermal cells. And then we get into the visible light spectrum. And for us, the visible light spectrum ranges between like 400 and 740 nanometers. So that's the violet spectrum all the way down to the red spectrum. Violet is a very, very strong wavelength, or a very, very strong photon, very short wavelength. And then reds have a much longer wavelength and represent less energy. And it's important for us to remember that this phenomenon that is color is a product of light reflectance, not light absorbance. Or really, I guess it's a combination of the two. But the reason that my shirt appears blue to you is because it's reflecting the blue light spectrum. And the reason that plants appear green is because they're reflecting the green light spectrum. What that means is they're not absorbing that particular wavelength or those wavelengths and converting that bit of the visible light into ATP and then ADPH and ultimately into sugar. So there are a couple of pigments in the plant, in the chloroplasts. And they are chlorophylls A, chlorophylls B, and then something called the accessory pigments. And this graph here shows you the absorbance spectrum of specific pigments. And you'll have to follow it along in your own copy of this or in your textbook. But it shows really three lines. It shows a solid line that represents chlorophyll A. It shows a short dashed line, which represents chlorophyll B. And then it shows a long dashed line, which represents the carotenoids. Now, along the y-axis, we have the amount of some particular wavelength that's being absorbed. And along the x-axis, we have the wavelength of light. So what you can see is, here at 400 nanometers, chlorophyll is absorbing quite a lot of that light. It's absorbing a lot of very high energy light, but then that absorbance drops off precipitously. And there's very little that's absorbed in the blues, greens, yellows, oranges, and then back in the reds, there is another peak. So it absorbs both very high energy and very low energy light. Chlorophyll B is very much the same, where you get a peak, a little bit longer wavelengths than chlorophyll A. So here in the blues, a peak in blue and then a precipitous drop. And then another peak again here in the oranges, a little bit shorter than the reds. The chlorophyll A was very good at absorbing. And then you have the accessory pigments that absorb in this range right here, the blues and in the violets, and a little bit into the greens. Those accessory pigments are the colors like yellow and orange and red. And those are the colors, the fall colors that we see. And really what's happening is plants are abandoning their chlorophylls because chlorophylls are also sensitive down here in low energy light. And the presumption is with higher energy you can stockpile more sugars. And so as we're approaching this dormant period that is winter, during the fall, they shift their absorbance spectrum to the high energy lights. They absorb a lot in here, really get photosynthesis going, and generate lots and lots of sugars. Okay, so I need you to understand the process of the light reactions and the architecture of some important structures. I would like you to know the architecture of what's called a photosystem. So a photosystem is the structural proteins that hold on to chlorophylls, and really it consists of two primary regions. And it looks like a canister with a very thick wall. So if I was to take that canister and I was to bisect it and then rotate it and show it to you like this, that bisected canister would have this shape. Where these are the thick walls that are going to come out and completely surround this central part. So these thick walls hold on to chlorophyll. So help me find a green marker. They hold on to chlorophyll. So interspersed throughout this canister, we have these chlorophyll molecules. During the first phase of the light reactions, what happens is a photon comes along and strikes a chlorophyll molecule, and then chlorophyll excitation occurs. That's where one of the electrons in the magnesium ion in the chlorophyll molecule gets so excited that it jumps out an energy shell. It absorbs that energy and it causes an electron to jump out. And that distance between the ground state and where it moves to represents energy. Ultimately, that electron is going to fall back to its ground state, and when it does that, it releases that energy, not the electron, but the energy and energy is passed between adjacent chlorophyll molecules. That's what we refer to when we say light capture. Now, ultimately, that light energy is captured and brought to what's called the reaction center core, and that's this central part right here. So this is reaction center core right here. And the reaction center core has a couple of really interesting molecules. The first of which is a large chlorophyll complex, and depending on the type of flow system that we're dealing with, it can either be called P680 or P700. We'll say this one is P680. And P680 receives that energy from these chlorophyll molecules over here. But P680 gets so excited, remember this is a chlorophyll molecule, and so you have electrons orbiting around magnesium ions that are doing funny things. It's so excited that it loses an electron. Now, when it loses an electron, it becomes oxidized. Remember, oxidation reactions are those in which electrons are lost. So P680 has just been oxidized, which is an incredibly unhappy state for P680. In fact, it's one of the strongest oxidizing agents in nature that we know of. So what it does is it takes water, H2O, and it tears it apart and removes one of the electrons from it. So that what we get is oxygen gas after some number of these water molecules have been torn apart, and then hydrogens that have lost their electron, or hydronium ions, or said differently, a proton. Because remember, hydrogen consists of just a single proton and a single electron. And P680 has taken that electron from that hydrogen, so it just becomes a proton floating around by itself. So water reduces P680. It's taken its electron, it's become reduced from an electron from water. And the electron that was a part of P680 goes to the top of the reaction center core to a protein complex called the primary electron acceptor, or PEA, primary electron acceptor. So that electron finds its way up here, and P680 reduces the primary electron acceptor because this electron acceptor has accepted that electron. It's been reduced. It doesn't hold on to it for very long. What it does is it passes that electron over to what's called the electron transport system, or the electron transport chain. The electron transport chain is a series of proteins that are used to create a proton gradient. So both the photosystem and the electron transport system, or electron transport chain, are embedded in the thylakoid membrane of the chloroplast. Like so. And this is the electron transport chain, or electron transport system, the ETC. So what happens is this electron is passed over here to this first protein. And that electron is accepted by that protein, and it's called a cytochrome protein. And that electron is passed from this first protein in the chain to the next one, to the next one, and then eventually it just falls out the bottom of the electron transport chain. Now, that happens for a specific reason. And the reason that it happens is this electron transport chain is going to do some work. And the work is powered by this falling electron. Think about it as an electrical charge. These cytochrome proteins become energized by accepting this electron, and they're able to accomplish something. And that thing that they accomplish is taking protons and pumping them from one side of the thylakoid membrane to the other. So these protons that are all being generated over here by the splitting of water end up coming over here and getting pumped through the membrane and out. Like that. Okay, so what we've created so far is a proton gradient. Lots of protons on this side of the membrane, very few protons on this side of the membrane. That's an unhappy thing, right? Those protons are naturally going to want to follow their concentration gradient and diffuse back across the thylakoid membrane. I'm going to blank out the screen and lift up the screen so that I can continue my drawing for just a little bit. What happens next is there is a protein incorporated into the thylakoid membrane, and this protein is called ATP synthase, and it has a single job. It just creates ATP. The way that it does that is by allowing these protons to filter back through the thylakoid membrane. It's like a channel for protons, like so. And these just fall back out. Now, every time a proton percolates through ATP synthase, it physically rotates in the membrane. So it spins in this direction, let's say. I don't know that it actually spins clockwise, but I'm going to say that it does. So it spins physically like that, it rotates. That's a kinetic form of energy. ATP synthase takes that kinetic energy, and then it takes an ADP, which is adenosine diphosphate, plus an additional inorganic phosphate, and to that it attaches, well, to that adenosine diphosphate, it attaches that inorganic phosphate to make adenosine triphosphate. Now, this is